Our next one is Paul Glue. He's the department chair and Hazel Buckland professor in psychological medicine at the Dunedin School of Medicine, University of Otago, New Zealand. I'm probably mispronouncing that dreadfully. His broad interests are in pharmacology, clinical trial design, and statistics, including trial stimulation and adaptive trial designs. And his research interests include the development of novel therapeutics and the treatment of mood disorders. Paul Glue.
Thanks. Okay, well, welcome everybody. I very much appreciate the opportunity to present some of my recent clinical research data with Ibogaine and Noribogaine. In New Zealand, we're not unique, but we're unusual in the sense that Ibogaine's been prescribable since 2010. The challenge has been that there are so many unknowns, though, that we don't know what the right dose is, what the right dose range is, how should the drug actually be administered, as one big dose or as multiple doses, how to monitor and manage safety. And if Ibogaine had been through a conventional drug development program, these issues would certainly have been addressed by the time it got to registration. Now, the perspective I'm going to bring today is based on having worked in drug development for 20 years in big pharma. And for those of you who are interested in any aspects of this topic, I published an article in Chacruna last week, which goes into this area in a bit more detail, and I do encourage you to read it. So, although Ibogaine has centuries of use as an ethnopharmaceutical, its published information on its clinical use is much more limited. We have early data from CBIG, who are part of Novartis now, who were looking at it as a possible agent to reduce opioid tolerance. We have Howard Lotsoff's serendipitous discovery that I guess is legendary now, and then in the 1980s he filed patents on using Ibogaine for interrupting addiction of a number of different sorts. We have studies from the 1960s where Ibogaine was being used to enhance psychotherapy. The NIDA did have a brief period in the early 90s where they were sponsoring a clinical trial of Ibogaine, but after a patient in the Netherlands died in 1995, they pulled the pin on this. So again, I'd like to contrast the estimates. I think Ken Alper had come up with numbers in the thousands, and it must be much larger than that by now, of people who've actually received Ibogaine for addiction interruption, against the relatively sparse published literature on using Ibogaine. We have a total of 7 case series. We don't have any randomized controlled trials. And if you look at the slide, you can see that the dose ranges go from a flat dose through 6 to 29 milligrams per kilogram, and it's been used to treat addictions to opioids, ethanol, and cocaine. In 2011, not 2013, I'd started discussions with Demorex, who are a small pharmaceutical company based in Florida. They have Deborah Mash works with them and is on their board of directors, and they'd obtained a patent for using Noribogaine in opioid withdrawal. Noribogaine is what Ibogaine gets turned to in the body. It's just demethylated Ibogaine. The alternative, which was to try and develop Ibogaine, was not seen in any way possible by Big Pharma, principally due to the fact that Howard Lotsoff had patented this use in the 1980s. The patents had run out, and there was no economic economic rationale for developing Ibogaine, because there would be no data exclusivity, that generics could come along and sort of take up the market once the drug was approved. And so the plan with Demorex was to run some pharmacokinetic safety and pharmacodynamic studies. So these are sort of fairly early phase pharmaceutical company trials studies of Noribogaine in New Zealand, and the main reason is that we had a very favourable regulatory environment, that Noribogaine was an approved drug. And so I'm going to be presenting data today from 3 studies. Studies 486 and 513 were both sponsored by Demerix, and the 486 was in healthy volunteers, 513 was in patients on methadone maintenance, and essentially what the design of each study was to look at ascending single doses of drug and monitor safety, pharmacokinetics, and pharmacodynamics. The third study I'm going to mention today is looking at the activity of an enzyme called CYP2D6 on Ibogaine pharmacokinetics. Now, CYP2D6 is the preferred enzyme for converting Ibogaine to Noribogaine. If it's inhibited, what are the effects of on Noribogaine and Ibogaine levels in man? So the 486 study, the one in healthy volunteers, we recruited 36 healthy male subjects. We had 4 dose levels, 3, 10, 30, and 60 milligrams. And in each dose level, we had 9 subjects, 6 on active, 3 on placebo. And because we used randomized double-blind dosing, no one knew what subjects were getting. We took blood samples to look at pharmacokinetics out to 9 days. We did safety lab tests, ECGs, looked at adverse events, and our anticipated side effect profile when we're designing the study was that this was going to look like a serotonergic opioid, sort of the love child of Prozac and morphine. And looking at opioid pharmacodynamics, if this drug were acting like a mu-opioid agonist, we'd expect to see a reduction in pain. And there are humane ways of assessing pain sensitivity. This is called a cold pressor test, and you stick your arm in a bucket of ice water for up to 5 minutes. And usually most people can't handle that. They pull their arm out before. And you can look at time to withdrawing of your arm from the ice water or pain ratings. And an opioid would be expected to extend the period of time you could keep your arm in the ice water. Mu-opioid agonists also can cause pupillary constriction, and this can be monitored very easily and quickly with a handheld pupillometer. And I would expect if Noribogaine was acting as a mu-opioid agonist that we'd see pupillary constriction that was higher at bigger doses. Mu-opioid agonists have an effect on respiratory drive, and that can be easily monitored with capnography. And then one can also model changes in whatever pharmacodynamic variable you're looking at against concentration and come up with exposure response modeling. These are the concentration time profiles for Noribogaine after the 4 doses. On the horizontal axis, we've got time in hours. On the vertical axis, we've got drug concentration. And one can see that as you push the dose up, you get a higher peak concentration. And if you model peak concentration or area under the concentration time curve, this is dose linear. So it's very well behaved. The more drug you take, the higher the concentrations. The half-life for elimination is around 30 hours. And we have a high volume of distribution. So a majority of the drug is actually sitting in body tissue rather than in the blood. And given that this is a very lipophilic drug, all of this is quite expected. When we're looking at safety and tolerability, there were no changes in lab tests or ECGs. The side effects were very mild. The majority of patients who reported them reported headache. And actually, we saw most of the side effects in the people on placebo or at the lowest dose. So certainly nothing that looked like a serotonergic opioid. When we looked at the pharmacodynamics, again, they were completely negative. We saw no change in pupil diameter between placebo and any of the dose groups. We saw no change in respiratory drive. We saw no change in cold pressor testing results. So we had to conclude this is not acting like a morphine, a mu-opioid agonist. And at the same time that we were mulling this over, Antonio, who representing a group of about 4 US-based laboratories, published a really detailed article in PLOS that Noribogaine was either an antagonist or weak partial agonist. So the problem we had there was that the best, or some of the literature up to that point, was very clear that Noribogaine was working as a mu agonist. The other thing that we were surprised not to see was that there was no nausea, anxiety, or insomnia. If I were to give you 60 milligrams of paroxetine, you'd see lots and lots of that. And we saw none of it. And then this Danish biochemistry group published, again, a very detailed paper showing that the way that Noribogaine binds to the serotonin transporter is quite different from SSRIs, that it binds in a very different conformational state. And my interpretation of that is it's not having the same effect on inhibiting reuptake of serotonin. The 513 study, this is the study in patients on methadone maintenance. We recruited 27 male and female subjects. They were on an average of 25 to 80 milligrams of methadone a day. And because methadone has a half-life of about 50 hours, and that might complicate interpretation of time to restarting opiates, we switched them to morphine, which has a half-life of about 3 or 4 hours. And that process occurred over a week before we were to dose them with Noribogaine. We had 3 ascending Noribogaine dose levels, 60, 120, and 180 milligrams, and only a single dose was administered. We had 9 per dose level, 6 on active, 3 on placebo, and because we were using randomized double-blind dosing, nobody knew whether people were getting active or placebo. We collected pharmacokinetic samples out to 1 week. In addition to the safety assessments we'd done in the previous study, we got Holter monitor ECGs on the day before dosing and on the day of dosing with Noribogaine. And we added 2 other opioid pharmacodynamic tests in, opioid withdrawal rating scales and time to restarting opioids. Oops, wrong one. So the first part, which was switching people from methadone to morphine, we managed to get all 27 patients through the switch. There are different guides on what sort of dose ratio should you look for if you're switching people from methadone to morphine. And the most common one is around 4-to-1 multiples. So if someone's on 30 milligrams of methadone, they need to be on 120 of morphine. We found that almost all of our subjects needed higher doses. We wound up with a 5 times multiple on average. And overall, it was pretty well tolerated. We got headache and nausea and constipation as the main reported side effects. These are the concentration time profiles after the single doses of 60, 120, and 180 milligrams of Noribogaine. Again, we saw dose linear pharmacokinetics. The higher the dose, the higher the peak concentration, and the higher the area under the concentration time curve. The time at which peak concentrations occurred was about an hour or 2 later compared with the healthy volunteers, and this most likely reflects an effect of morphine given pre-Noribogaine on the speed of gastrointestinal transit. We had the same sort of estimate of half-life, somewhere between 24 and 31 hours, and a very high volume of distribution, again, showing extensive distribution into tissue. Time to restarting opioids. It has to be pointed out that this was not a primary endpoint in the study, but if you look at the top table, you can see that there's nothing that looks like a dose response. We took almost 14 hours for the people in the placebo arm to restart opioids. We seem to perhaps have something at 120 milligrams, but at 180 milligrams, I would expect there to be an even greater prolongation before starting opioids, and we saw nothing like that. So in terms of a dose response, this is meaningless. If you look at the bottom panel, which breaks out the data from the the placebo arm associated with each dose cohort, you can see that actually there's a very close association between how long people took on placebo for that cohort to restart opioids and those in the Noribogaine group. And a Pearson's correlation is highly significant. So it seems as though the time to restarting opioids is very much confounded by when the people in the placebo arm wanted to restart opioids. The clinic we were testing people in, we had fairly intensive telemetry going on. And when somebody first put their hand up and said, I'm going into opioid withdrawal, my pupils are getting bigger, I'm starting to sweat, that seemed to influence other people into then saying, me too, I want to restart opioids. So, bit of a problem. Yeah, they're in the same room. Again, it has to be pointed out that the key elements here were safety and pharmacokinetics. You know, that this is a less than ideal design for monitoring time to go into withdrawal. In regards to safety and tolerability, the most striking thing were the reports of changes in light perception, that the light seems different, the light seems brighter, colors seem brighter. This occurred within an hour of dosing, and it had finished by 2.5 to 3 hours after dosing. And there seemed to be a dose-effect relationship in that it was only seen at the 2 higher dose levels. Looking at the timing here, the Noribogaine didn't achieve peak levels until about 4 or 5 hours after dosing. So it's interesting that it came on and also disappeared during the ascending or absorptive phase of the pharmacokinetic profile. We didn't see anything that looked like a hallucination. Perhaps the most important finding are the changes in heart repolarization. Now, for those of you who are not cardiologists, in the top left-hand panel, you'll see an ECG complex. The tall, pointy part of it is called the QRS complex, and that's a sign that the ventricle is depolarizing, i.e. it's pumping blood around the body, and then the bump to the right of that is called the T wave. And that's a sign that the ventricle is repolarizing, so it can go through the same cycle in a second or so later. The QT interval, so from the start of the QRS complex to the end of the T wave, that's the thing with the red circle around it, varies according to heart rate. And so the way we adjust the QT according to heart rate is we do what's called a correction. We divide it by the cube root of the RR interval, or the number of beats per minute. And prolongation of QTC is significant in that it's associated with arrhythmias, and not nice little arrhythmias, ventricular arrhythmias such as torsades de pointe that are associated with death. And graph A shows the relationship between the Noribogaine concentrations for all of these methadone patients. On the horizontal axis and on the vertical axis, we've got changes in QTC. The dotted line in the middle of that graph is at 10 milliseconds. Normally, FDA or other regulatory agencies start to get squirmy when they see an increase of more than 5 milliseconds. So what we're seeing at the highest dose, 180 milligrams, at peak concentrations, we're getting on average a 42 millisecond increase in QTC. The graph B is simply modeling of the data, and so we can come up with estimates at peak concentrations. Either way, this would be seen as a significant safety signal. It doesn't mean that Ibogaine or Noribogaine would be undevelopable, but it does make it more challenging. One would want to have a lot of cardiovascular safety assessments in a clinical trial. The last study is looking at the metabolism of Ibogaine. Ibogaine is on the left, and you can see it differs from Noribogaine on the right by a methyl group, a CH3 group. And 2D6 is the enzyme that primarily catalyzes that. There are a couple of other enzymes, 2C19, 3A, but they're slow and inefficient. We know that 5 to 10% of most populations have genetically absent 2D6 activity, and some drugs like Prozac and paroxetine inhibit it as well. And a research question was, what's the influence of inhibiting 2D6 on Ibogaine pharmacokinetics? So we had a rather complicated study design. On day 1, we gave everyone a dose of Robitussin, dextromethorphan, and collected urine for a few hours afterwards to look at the metabolite of look at the ratio of dextromethorphan to its metabolite dextrorphan, and that gives you an index of 2D6 activity. We then put people on either paroxetine, which is a 2D6 inhibitor, or placebo, gave them another phenotyping test on day 7, and gave them a dose of Ibogaine on day 8, 20 milligrams single dose. These graphs show the change in dextromethorphan to dextrorphan ratio in the people who got paroxetine. You can see that on day 7, their ratios have all gone up, suggesting that their 2D6 activity is much less. And the people who got placebo, the graph on the right, show no such change. This is the effect on pharmacokinetics. So the graph on the left are from the people who got placebo, so normal 2D6 activity. The tiny little red blob in the bottom left-hand corner is the Ibogaine. So it's barely detectable for perhaps 2 or 3 hours after dosing. It's all being converted incredibly rapidly to Noribogaine, which is the black line in the left-hand graph. On the right-hand graph, we've got the data for the people who got paroxetine pre-treatment, who've had their 2D6 activity decreased. You can see there's a gigantic amount of Ibogaine, peaks at around 2 or 3 hours, but it's still detectable out to 72 hours. Now, at the same time, you're seeing Noribogaine levels, but the fact that the Ibogaine signal is so great at that point that essentially when we try and add the Ibogaine and Noribogaine concentrations together, and we can look at molar concentrations and calculate active moiety, you get about a doubling of exposure to both Ibogaine plus Noribogaine in people who have reduced 2D6 activity compared with people with normal activity. And bear in mind that we have substantial safety issues associated with those very high blood levels of either Ibogaine or Noribogaine. We didn't see any effect on psychometric tests. We didn't see any increase in ratings on being more alert or energized. So trying to sort of sum up the things that we were expecting to see and didn't see, we were expecting an effect on QTC, but certainly not as profound as what we'd identified. And it certainly limits what blood levels or dose you might want to try in further efficacy trials. 2D6 has a huge effect on Noribogaine levels. Then neither of the drugs are mu-opioid agonists. They don't have SSRI-like side effects. They don't make you feel more alert or energized. And although this is a much less than desirable study design, we couldn't show an effect of Noribogaine on time to restarting opioids. So the unresolved issues for me are, what is the overall effect of Ibogaine and Noribogaine and opioid withdrawal? I don't think that the 513 study was a definitive negative study, but even if it does work, the question about dose and achievable blood levels is going to be really important. And ultimately, this may have to be driven by effects on QT intervals. The other big challenge is that some of the older data, the pharmacology, the behavioral effects appear to be unreliable. So if you're an Ibogaine provider, if you're thinking of dosing patients, you need to have really good ECG monitoring, not just a pulse meter, but you need to have people in some sort of telemetry for the first few hours after dosing to look for the presence of arrhythmias. You need to have an IV line in, you need to have access to magnesium or a defibrillator if people do go into a serious arrhythmia. It's unclear what a safe Ibogaine dose is, and I think Claire is going to be talking on using repeated low doses later today. From a pharmacokinetic perspective, I'd say that makes a lot of sense. And then finally, if you're going to be dosing patients, know what their 2D6 activity is. You can get genotyping easily. There may be phenotyping is complicated and expensive. if they're on 2D6 inhibitors, you need to take them off that. And I'd like to say, finally, thank you to my collaborators. Thank you very much. Thank you.